Good morning, dear students. Today we are going to start a new chapter, that is evolution of developmental patterns in unicellular protist. For understanding cell differentiation and the process of evolution of sexual reproduction, in the early period onwards, scientists selected protists, unicellular protists as the model organisms. It is very easy if consider such type of organisms. The first point that we are going to learn is control of developmental morphogenesis. In that, what is the role of nucleus? What is the role of nucleus in the development of morphogenesis that we are going to see now? The first proof that the nucleus contains the hereditary or the developmental information. It assembled because of a certain type of experiment. For all those experiments, scientists took simple unicellular organisms and they fragmented those organisms. We know that as a unicellular organism is fragmented, some fragments will get a portion of nuclear material and some didn't get any portion of nuclear material. And those fragments, that is carrying the nuclear fragments, nuclear fractions, are able to regenerate and to form the entire organism again. That proved that nucleus carry the hereditary or developmental information at first. Okay, and next we are going to see what are the specific experiments done by scientists to further substantiate that findings that nucleus itself carry the hereditary or developmental information. That is what study of cell morphogenesis using acetabularia. Acetabularia is a unicellular green algae. Even if it is appear to carry so many structures, it is a unicellular algae. It consists of three parts. Which are the parts? A cap, a stalk, and a rhizoid. This is the cap, this is the stalk, and this is the rhizoid. And this rhizoid portion helps the plant anchored to a substrate. And what's the experiment and who is the scientist performed that experiment? It is Hammerling. And what was that experiment? He considered two Acetabularia species, that is Acetabularia mediterranea and Acetabularia granulata. What he did was, he exchanged the nucleus of these two species between them. The nucleus of this Acetabularia mediterranea is transferred to Acetabularia granulata, and from here the nucleus is transferred to this. And as an example, as we take the case of one progeny, we can find that here the stalk belongs to Acetabularia mediterranea, but that cap belongs to Acetabularia granulata. Why? Because here the nucleus comes from Acetabularia granulata. So, as the nucleus determine the type of the cap, here the cap formed is that of Acetabularia granulata. Okay. In that way, they found that nucleus carry the hereditary or the developmental information. And the next that form for understanding the process of formation of cap. That formation is a complex morphogenetic event. For synthesizing or forming a cap, it need to synthesize numerous proteins. And that formed proteins need to accumulate in some portion of the cell. And that may later lead to the formation of cap. The fact that the information responsible for the formation of cap, it is accumulating in the cell. It is proved after the scientists perform some more experiment as a continuation of the first experiment. What is that? I will explain. I told you that in the first experiment, Hammerling transferred the nucleus from Acetabularia mediterranea to Acetabularia granulata. I'm going to draw the picture of Acetabularia mediterranea. It is carrying a round cap, and the scientist transferred its nucleus to Acetabularia granulata. Acetabularia granulata. Now it is carrying the nucleus of Acetabularia mediterranea. And the expectation is that as it is carrying the nucleus of Acetabularia mediterranea, it need to form the cap that of Acetabularia mediterranea instead of Acetabularia granulata. Okay. But after transferring this nucleus in its early developmental period itself, again scientists removed this nucleus from here. Even then, it formed a cap that of Acetabularia granulata. Okay. After a period of time, this Acetabularia granulata dies off because it didn't have a nucleus. That is the reason. And as a result of this experiment, scientists understood that the information responsible for the formation of cap had reached the cytoplasm long before the formation of cap. And they arrived in these two notable findings, that the nucleus contains information specifying the type of cap to be produced. That is proved. And also the material containing the information for the cap formation. It enters the cytoplasm long before the cap production. That two things they find out. For explaining these two notable findings, the scientists proposed a hypothesis. That hypothesis is that the nucleus is forming a stable mRNA, and that mRNA lies dormant in the cytoplasm until the time of cap formation. And they proved that explanation through an experiment. And that experiment is again done by Hammerling. What was that experiment? They fragment the acetabularia. That's what I am going to explain. This is the acetabularia, and he fragment this algae into several pieces. And this piece we know is a base piece, and it is carrying the nucleus. And after a period of time, he noticed that the fragment that is carrying the nucleus give rise to entire green algae, and another piece that means the tip portion, that fragment also give rise to the entire organism. From that finding, he proved that the information responsible for the cap formation, it is stayed dormant, and it is stored in the tip portion till the time of cap formation. It is proved. Along with that, scientists understood the significance of cytoplasm. The significance is that even if the mRNA is present in the cytoplasm for a long time, its expression doesn't happen there. That means some factors that present in the cytoplasm keep that mRNA or prevent the translation of the mRNA till the time of till the proper time of cap formation. So, in that way, cytoplasm also playing a leading role in the formation of cap. Okay, then what is the ground level? What is the underlined finding they arrived here? It is that the development is controlled at both transcriptional, that is the nuclear level, and also in translational level. That means some factors present in the cytoplasm is regulating the translation of the mRNA. That is the final finding regarding the development arrived by the scientists. Okay, I think it's clear to you. With this, we finish today's section. Thank you. I'm an icon, man. Ain't my girl about a 10. I should fuck her in a Benz.